Hello, my name is Robert Foster. I'm the CEO of Hepion Pharmaceuticals, and we're headquartered in Edison, New Jersey, as well as we have an R&D facility in Edmonton, Canada. We're a company that targets liver disease, and more specifically, we target the development of our lead molecule, CRV431, in a disease called Nash. Nash, if untreated, can lead to liver cirrhosis, hepatocellular carcinoma, we call it HCC, which is liver cancer, and it can also lead to death. There are no FDA-approved medications to treat Nash right now, and Nash is a big burden on the healthcare system. Nash is actually a more severe form of fatty liver disease, which affects about 1 in 4 people on planet Earth. The number of people that have Nash in the US is about 17 million people, so it's very prevalent. This particular page has 2 panels. These 2 panels talk about the regulatory endpoints when it comes to Nash drug development. One of those endpoints at the top left-hand side of the page is called Nash resolution. The other endpoint is called fibrosis improvement. And what you can see is that if we look at the placebo response, you can see a fairly low response, whether or not you look at Nash resolution or whether you look at fibrosis improvement. There's a low response for placebo, as you would expect, but nevertheless, there is a response. Now, if you look at the red lines for both of those panels, you can see that's the response you get at high dose of the drugs that I've outlined on the left-hand side of each of those 2 panels to show at a high dose of these drugs, what is the response according to these 2 endpoints. And you can see that the responses are quite low, 27% and 24% for Nash resolution and fibrosis improvement, respectively. So the spread or the delta between high dose and placebo is actually quite poor. So what we need to do as an industry is to find out ways of developing drugs that we can have a better response. And so I believe that we need new therapies in development for Nash treatment that show a much better response. And that's what Hepion is trying to do. If we move now to what it is exactly that CRV431 does, CRV431, I'll call it CRV, inhibits certain cyclophilins. Cyclophilins are enzymes in the body that can regulate a number of different processes, and these are mostly all related to the way proteins are folded in the body. But to really drill into it a little bit more in detail, I can say that there are 17 different types of cyclophilins. We call these isoforms. Here on this page, I've outlined 3 important cyclophilins. There's cyclophilin A, cyclophilin B, and cyclophilin D. So just a few words about each one of these cyclophilins. Cyclophilin A has a role in inflammation. And if we block the role of cyclophilin A by CRV431, we can inhibit inflammation. If we go to the next cyclophilin, cyclophilin B, we know that cyclophilin B is key, and it's actually very important in the production of collagen. Again, collagen being the protein that causes the fibrosis or the scarring of the liver. We can inhibit cyclophilin B with CRV. And then finally, the role of cyclophilin D. We know that cyclophilin D is involved in cell injury and cell death, and therefore if we inhibit that particular cyclophilin, we can protect cells from injury and death. And that's, again, those 3 key components of what CRV does. So how did we come up with CRV431? We started with a molecule back in the 1980s called cyclosporin. And cyclosporin is a great molecule that completely transformed the way organ rejection or organ transplantation was dealt with. Cyclosporin is a cyclophilin inhibitor, but it also inhibits the immune system. So it's an immunosuppressive drug. And cyclosporin has a number of drawbacks. So in the mid-1990s, I struck out and developed a company starting in 1993, which today is called Hepion Pharmaceuticals. And what we did back in those days, in the mid-1990s, is we developed what I would call a new improved version of cyclosporin, which is called voclosporin. Voclosporin is also an immunosuppressive drug, and today's valuation of voclosporin is approximately $4 billion. And then what our team did back in the mid-2000s, probably about 2007, 2008, is we stripped away the immunosuppression of cyclosporin and voclosporin, and we developed CRV431. We also developed a library of about 200 other molecules, and the key point here is that these are cyclophilin inhibitors, but they are not immunosuppressive, and therefore we can use these molecules to address a number of different things, which I've outlined at the bottom of this page here. The investigational new drug applications that we've submitted for CRV431 are outlined on this page. So the key IND that we have underway right now and a key indication that we're working on is Nash. We are looking at enrolling our first patient in phase 2B in early 2022, and we're looking at submitting our new drug application to the FDA probably no later than 2028. We do have a couple of other INDs, but we're not pursuing these at the moment unless we get external collaboration or partnerships with other companies. If I now move to the next page, which talks about our non-clinical or preclinical studies, we've done quite a number of studies. And the question is, why did we do so many? Well, what we wanted to do was to pressure test CRV431 in as many ways as we possibly could, so that before we get to the clinic and spend a lot of money to develop this drug, we knew absolutely for certain, or as certain as we can be, that CRV is a good molecule to be pushed into the clinic. So the first thing we did is we looked at in vitro studies of CRV431 just to make sure that CRV had an effect on the generation of collagen. And we successfully did that in what we call an in vitro study. We then moved to a number of different preclinical models of Nash. These are in vivo studies. And we did about 8 or 9 or so models of these Nash animal models to show that we had a consistent and a very potent inhibition of fibrosis. We showed this repeatedly. And we did this globally. We did studies in Japan, we did studies in France, studies in various locations in the US, and we consistently saw from lab to lab that we had an antifibrotic effect. And finally, we did a study in the UK wherein we took human liver slices and we poked those slices in a way, or we stimulated these slices to become fibrotic. Then we showed that CRV could actually inhibit the fibrosis of these human liver slices. And taking all this together, we then decided, okay, we've got a molecule that we can move to the clinic. So exactly what are we doing in the clinic? Well, I'll go through our phase 2A study, which is called the Ambition study. The Ambition study was a phase 2A study, which was placebo-controlled. We took 2 different doses of CRV431, a low dose at 75 milligrams, and a high dose at 225 milligrams. Then we gave each of those 2 doses as well as placebo to subjects that had presumed F2 and F3 fibrosis. We studied administration repeatedly, once a day, orally, soft gelatin capsules of CRV431, and that was given for a total of 28 days, so 4 weeks, and then we did a 2-week follow-up. We wanted to assess at least that the drug was safe, well tolerated, and we also wanted to characterize the pharmacokinetics. Moving now to the next page, which addresses the patient demographics, probably there's 2 key points to make here. One is that these people had a historical biopsy that indicated they had F2 and F3, but certainly they were in the right age range for the presumed F2 and F3 fibrosis. They also had the right type of body mass index, so these generally higher BMIs than normal subjects. If we now move to the next page, which talks about some of the primary endpoints, first of all, I'll talk about the exposures or the pharmacokinetics of CRV431. There's 2 figures here. On the left-hand side is day 1, pharmacokinetics. On the right-hand side of the page is day 28, pharmacokinetics. And the red line talks about the 225 milligram PK profile or pharmacokinetic profile. The blue is the low dose or 75 milligrams. What we can see is that the blood concentrations of CRV431 quickly got into what we would consider to be the efficacious range. And we do know that the efficacious range is probably north or higher than 800 nanograms per mil. So we quickly get into that range in either the left or the right panel, but it's a little bit slower if we look at the 75 milligram single dose on the left-hand side. But by the time we get to 28 days, we're certainly above that 800 nanogram per mil range. So either one of these 2 doses look to be probably in the sweet spot when it comes to efficacy and how we're treating these subjects in the future. Now, Because these were F2 and F3 NASH subjects, we also wanted to look, in addition to the safety, tolerability, and PK, we wanted to look at, is there any way we can assess or get an early indication of efficacy in these NASH subjects? So one of the things we looked at was ALT. And again, I mentioned that if we inhibit, for example, cyclophilin A, we should inhibit inflammation. One of those markers of inflammation is ALT, which is a liver enzyme. So looking on the left-hand table, we can look at ALT starts to decline. So if you look at placebo versus 75 versus 225 milligram dosing, you can see again, on average, that you get a dose-dependent reduction in ALT, going from ALT drop of about 6.1% down to about 18% and then 21% going from placebo to high dose. Similarly, we also see that the number of subjects goes up in ALT response as the dose goes up. So we see that about 50% of the placebo subjects had an ALT decline, and then up to about 87 to 90% of those subjects showed an ALT decline as you get to the high dose. Interestingly, again, placebos given to subjects actually do see a response in most clinical trials, and that is because it's well known that if you lose weight or if you watch your diet, you can actually get a response, and that's what we call the placebo response. The graph on the right-hand side of this page also shows that you can see that there's a dose-dependent reduction in ALTs. If we move to the next page, we can look at, is there a way that we can link the pharmacokinetics, in other words, the blood concentrations, to the pharmacodynamics, which is, for example, the drop or the decline in ALT response. And there is a way, and this is something I think that is quite unique to Hepion's approach to drug development. And similarly, we use this approach in our last company, Vitro Pharmaceuticals, to link pharmacokinetics to pharmacodynamics quite successfully. So here what we're showing is that there's a link between the pharmacokinetics and the pharmacodynamics. And so why is that important? We can link the PK/PD to predict, based on a blood level at a certain point in time, what will happen to the ALT decline in the future. And the right-hand graph shows the predicted versus the observed drop in ALT based on pharmacokinetics. You can see there's almost a perfect correlation between predicted and what was observed. We've also used machine learning, and we're employing that in our clinical trials, not only in a phase 2A, but also as we move forward in phase 2B and phase 3, we're using machine learning and artificial intelligence, or AI. This is a quick example of the supervised machine learning where we're looking at ALT responder analysis. So we're looking at a multivariate approach where we look at patient characteristics, and then we try to look at what is the predicted ALT and what is the ALT response. So on the left-hand panel, you can see that the number ones are the non-responders for ALT decline. The two are the responders. You can see that there's a clear delineation between the responders and the non-responders, and this type of analysis will help guide us through our upcoming clinical trials. We're also looking at what sort of biomarkers can we look at to discern what is going on with these subjects in their collagen or their fibrosis formation. And here we're looking at specifically something called ProC3. You can see, especially at the 225 milligram dosing, that there's a decline in the ProC3. This decline is observed in only 28 days for the 225 milligrams, which is quite remarkable because I have not seen any other study where you get a ProC3 decline that rapidly. And interestingly, the decline continues up till day 42, even after the drug administration has been stopped. So what that is telling us is that there seems to be an overlap or residual activity of the drug. And we know that the pharmacokinetics is telling us that the terminal elimination half-life of this drug is long. So therefore, it makes entire sense that the drug has a lag effect so that there's a continuous decline in the ProC3 even up until day 42. We've also similarly linked the pharmacokinetics to the pharmacodynamics, and the same holds true. We can take the pharmacokinetics, or we can take a blood concentration early on, and we can predict in the future what type of a ProC3 decline we should see. So again, the observed versus the predicted decline in ProC3 is incredible. And this is something that I think, again, is unique to Hepion's approach. We've also done the responder analysis. Again, this is machine learning, and we're doing a discriminant analysis, and we're looking at responders versus non-responders. And this is, again, shown on the panel on the left-hand side, which shows that there's a way to discriminate between responders and non-responders. Again, we've used it for phase 2A to look at our data, but we'll also be using this in our upcoming phase 2B as well as the phase 3 programs. I did touch on the AI that Hepion's been developing. So this is a proprietary approach to what we're doing, and we do have a clinical pharmacology team that's been working on AI since the 1990s, and we've also employed some of this previously in our last company, again, Hepion Pharmaceuticals, but now more honed skills that we're bringing to Hepion with our AI. So first of all, AI is allowing us to really understand what Nash is. So Nash is very, very heterogeneous. So we're trying to wrap our arms around what exactly is Nash and what are the different stages, if you want to call it, of Nash, and try to figure out what's going on. We're also trying to look at what are the biomarkers that we can look at to really get a handle on what is happening in Nash. So to give you a snapshot of one of the things that we're doing on what we call our multi-omic analysis, and what I mean by multi-omics, I mean genomics, I mean proteomics, I mean lipidomics, and metabolomics. So here I'm looking at genomics specifically, where we've done RNA sequencing. On the left-hand side, you can see there is sort of an ability to look at these reds and greens of something called a heat map, where we're looking at differentially expressed genes. And these are genes expressed at day 1 and day 28. And now if we look at a responder analysis of these subjects on the left-hand side, and we tease out some of the patient characteristics that we think the AI is telling us to look at specifically to be able to discern who is responding versus who is not responding so well to CRV431, you can see the figure on the right-hand side. All of a sudden, the heat map looks a lot more clear, where you can see the greens and the reds popping out. So the red is just telling us that these genes are now becoming more expressed or overexpressed, and the greens are becoming less expressed. So it's really telling us that the AI is really, really working to help us understand what's going on with these genes. Going to the next page now, we can see that these genes are part of a regulatory network. So these genes are talking to one another. And we've actually been able to use our AI to figure out which genes are talking to which genes, and genes that are maybe talking to proteins that are talking to genes. And in the left-hand side of this particular page, we're talking about how we color code. So we can look at, for example, collagen binding. We look at, for example, collagen catabolic processes. And we're looking at how all of this is sort of regulated. And we're looking at CRV431 as almost like a master modulator of this gene network. We've gleaned so much information of our phase 2A study using our AI. And again, we'll employ that for the phase 2B and the phase 3 program. So in conclusion on the phase 2A, we've shown that the phase 2A showed us the CRV was well tolerated, safe, and it showed us also the pharmacokinetics. It showed us that in only 4 weeks, we saw a decline in things like ALT and ProC3. It showed us that the AI is working quite well. We're able to pop out these heat maps in a way that we can discern which genes are sort of involved in what's going on with CRV and NASH. We're also able to see that, for example, 800 nanograms per mil seems to be the point at which we start to get an exposure that results in efficacy, and we'll employ that again as we move forward. So as we move forward into phase 2B, the Ascend NASH trial, here the primary objective is to look again at safety and efficacy, but now we're looking at 3 different doses, 75, 150 milligrams, and 225 milligrams. Again, this is once-day oral administration of a soft gelatin capsule. We'll be looking at administration of CRV for a total period of 12 months. This will be placebo-controlled, and we'll have a 1-month follow-up. This is a larger trial of F2 and F3 patients or subjects. These are biopsied subjects, biopsy before the trial, biopsy at the end of the trial, and we'll have a couple of interim analyses in there. And of course, we'll be looking at these biomarkers as well as the biopsies throughout this trial. This will be 336 subjects in total. So the primary endpoints, again, safety, tolerability, and PK. We'll be looking at Nash resolution or the reduction of fibrosis by at least 1 point. And then we've got a number of secondary efficacy outpoints that are also outlined on this page. So in 2021, the anticipated events, some of these have already occurred, of course. We're a good portion all the way through 2021. So we have completed the phase 2A program. We've completed our longer-term animal toxicology studies. We actually can see that we have no toxicology signals whatsoever on these animals. So this is a very, very safe and well-tolerated drug. And again, it's been borne out by our clinical program so far. We're continuing to scale up and look our chemistry and manufacturing, which is optimized. We're doing our chemistry manufacturing over in Europe. We've completed some drug-drug interaction studies. And again, we prepared the ground for the phase 2B to kick off with dosing of our first subject in Q1 of 2022. And the AI, it's a continual process where we're constantly honing the AI, and that's shown at the bottom of this page. Moving to the summary slide now, CRV431 is a once-a-day oral medication that hits several cyclophilins and therefore is a multimodal type of molecule. It's an antifibrotic, anti-inflammatory, it's also cytoprotective. So far, we know CRV431 is safe, well tolerated. We've easily characterized our pharmacokinetics. We've seen efficacy and efficacy signals in as little as 4 weeks. Again, the phase 2B activities are kicking off. Our core scientific team has at least a cumulative 100 years of experience, and I would venture to say that we're probably the most experienced cyclophilin team on the planet. We've been doing this a long, long time. We've got intellectual property that is robust. It's going to take us out quite a number of years. And in summary, you can also say that in addition to having these two value drivers, one being our drug molecule, CRV431, but also our AI, we do have a strong balance sheet. Our last Q filing showed that we had 110 million on the books, and we have 76.2 million shares outstanding. So I thank you for your attention and look forward to a bright future at Hepion with CRV431. Thank you.